Microscopy is as important today as it was when the first microscope was invented more than 400 years ago. In the Optical Imaging Facility in USC's Stem Cell Research Center, scientists use a specialized type of microscope, known as a confocal microscope, to visualize the spatial relationships amongst cells and sub-cellular structures, and to observe the movements and behaviors of living cells within organisms and developing organ systems. For example, to study kidney development, researchers label each progenitor cell type with different fluorescent antibodies. An objective lens captures the emitted fluorescence and magnifies and resolves the details of the labeled cells, revealing the intricate structures of the developing kidney. These microscopes are the visual workhorses for studies of stem and progenitor cell biology and developmental processes more broadly, and the high-resolution imaging of biological and disease processes within the cell. Through optical imaging, scientists gain the knowledge of biological processes that is critical for informing and advancing new treatments to improve human health.